This is a field disease problem. I visited about 20 fields, and 9 out of those 20 had hemp leaf spot in different regards. Usually, though, this disease, when it's there, it's really severe. So this is pretty much an example in this image of what it looks like in the field. So symptoms will start as early as like July 1st. By August 1st, you're really noticing it. Okay, so we can look at it a little bit differently. All right, yield losses up to 100%. We've had growers who've destroyed entire fields because it was so bad. It turned into a blight. I've got lots of pictures. And we know that at least 2 neighboring states have it. One of them is New York. So it's not Kentucky specific as we once thought it was. So it's not Kentucky specific. And as far as we know or knew, hemp only. We don't know where else it is. Well, we did. I'll get to that. All right, so here are some images. Wow, the lighting is really bad. So here are some images of the leaves and some close-ups. You'll see that these are round spots. They don't get any bigger than about 2 millimeters. Some of the spots will kind of be like a white with a tan border. Others will kind of be brown with a little bit darker border. When they get bigger, it's because the spots coalesce. And if you look at the centers of some of these spots, you'll see some kind of black dots. Keep your eyes open for that one as I switch slides. Ah, there we go. Now, no sleeping, because I can see you. All right. All right, so here it is in the later stages. So you'll see this blighting. As those spots coalesce, they're causing plant tissue death. So this necrosis is really destroying leaves. One thing about this one is those spots are not just on the top side of the leaf, but they're also on the bottom side. And those spores are on both top side and bottom side of the leaves. So it's pretty bad. All leaves seem to be infected. Like when we see infection, it's everything. So it's new leaves and old leaves and upper leaves and lower leaves. It's darn everywhere. It's evenly distributed through the field. Usually that's not typical of a disease, but it's everywhere. Here's a close-up. Now you can see this with the naked eye. You see those little black hairs sticking up? Those are the conidiophores. And so techie word for those are the little stalks that hold the spores on top. And they're that big and they're that dark that you can actually see them. So let's talk about the fungus that actually causes hemp leaf spot. So the genus is Bipolaris. Those of you, anybody here grow row crops, you know Bipolaris, right? So Bipolaris, the species is something we didn't know. We've been working on that. So a little bit about the Bipolaris, some kind of nerdy plant pathology stuff that matters to you. It creates a very large spore. What does a large spore matter? Well, large spores don't become airborne really easily, do they? They don't fly around like a small spore. So the macroconidia, in theory, they shouldn't move very far. It also produces, though, a microconidia, a very small spore. Those we think could be airborne. Is that how this thing is moving? And then it produces something we think is either a sclerotia or a protoparathesia. The sclerotinia would be a survival structure. It would fall on the ground and survive 6, 8, 10 years in the soil, if in fact that's that structure. If it's a protoparathesia, that means there's sexual recombination going on. And why does that matter? Mutations, right? So you've got adaptations happening. We don't know if that's what's going on. But that really matters in the way we approach some of our recommendations and where we go next with managing this disease. So here's some more nerdy pictures. So on the left side, you see the little skinny stalks and the big fat spore on top. So the little skinny stalk is simply just a stalk that's holding the spore up above the surface of the leaf. And there's the spore, and they germinate from all of the sections within the spore. So if anybody is into microbiology of any kind, you know, that's pretty cool. All right, there's the microconidia. The microconidia are what we suspect might be going airborne. We're not real sure. And on the right, those are those, whatever those structures are, the protoparathesia or the sclerotia. Those are the ones, you see how they're black and they're melanized. So in theory, anything that's black and dark and melanized is cold tolerant, UV resistant, fungicide resistant, heat resistant. So melanization is a protection, just like when our skin melanizes. So a melanized survival structure means this guy can probably hunker down and survive from season to season. And if that's the case, you as a grower should worry. So, all right. So what the heck is this fungus? So you don't have to know phylogenetics, I promise you. But we had to turn to molecular identification and actual whole genome sequencing to be able to figure out what this thing was. And I'm going to show you just a couple of things. So in this tree that you can't read, so this is like the tree of life. So think about a family tree. Who branches to what? This is a tree of the bipolaris fungi. And you'll see that the yellow ones are our isolates from Kentucky, our bipolaris, or our hemp leaf spot isolates. But where they're seated, it's clearly in the bipolaris, between Bipolaris oryzae and Bipolaris matus. So what does oryzae mean? What does matus mean? It's rice and corn. Bipolaris of rice, Bipolaris of corn. Okay? So we know about other Bipolaris species, especially the ones closely grouped in that tree that I showed you. Corn and rice and cereal grains, grasses, bananas and coconuts. Those diseases, or Bipolaris of those diseases, are very closely related to ours. Well, what's something about these that's different from hemp? They're monocots. They're grasses. Hemp is not a monocot. This makes no sense. So we continue to scratch our heads. So off we've been since 2015. We have been sampling weeds and plants in and around hemp fields. What else could this be on? My graduate student, Desi, if anybody has met her, she goes to your field a lot. If she goes once, she goes a lot. And she is sampling. She's sampling anything with a spot in and around hemp fields that have hemp leaf spot. And there are lots and lots. I just have a snapshot here of some things that she's been looking at. So she's trying to find an alternative host. What else is this pathogen infecting? And how does that connect to our hemp infections? She found 4 weeds. She found false nettle, hophorn bean copperleaf, velvet leaf, and Virginia copperleaf, all infected with our bipolaris. None of the grasses, none of the monocots, just these 4 dicots. So our big question next, though, is what is that connection? Was the hemp infected first and the inoculum level was so high that it bombarded the weeds around? Or is it that the weeds were infected and served as a reservoir and transferred this fungus to the hemp fields? That part we don't know yet. That's one thing that we are working on. All right, so our project continued, and we selected isolates or samples from 9 counties, and we are working with these 9. So in order to get a little bit deeper, we have to be able to compare the bipolaris isolates from the different areas. Are they the same? Or are they related? Okay, what's the variation going on there? So that's what we've been working on. We've been doing some whole genome sequencing to be able to get down to the nitty-gritty of just what is the relatedness of all of these different isolates. So here's another tree of life, if you will. And we've got our isolates there. I zoomed in a little bit. And I just want you to think about, so if all the yellow lines are our isolates from Kentucky, look how they're branching. They're not all the same. They're not all branching together. There's variation within that branching, which means they're not related. Just like you and I are not related. We're the same species. We're Homo sapiens, right? But we're not related. These things are not related. So now what? Is there sexual recombination? Why is there so much variation within this small area? The variation means something. Probably that these isolates have been separated from each other for many, many generations. So it's there. It's established. Wherever it's coming from, it's established. And I think that's a really important message, is that as hemp comes in, pathogens that once didn't matter to our typical crops are rearing their ugly heads. And this one is definitely rearing its ugly head. So, unrelated populations. What does that mean to you? Well, a lot more questions again than I have answers. What's the source for inoculum? Where is this fungus coming from? We just don't know. We absolutely don't know. What's the importance of the relatedness of these populations? How far removed are they from each other? We think they're all the same species, and we were at a point where we thought maybe there are two different species at play here, but we think they're the same species. And we just don't know what the primary host is. We absolutely don't know. One thing we do know is they're not transmitted by clones. If you buy a flat of clones from someone, they're not moving that way because it's not going in the greenhouses. In fact, we have a grower who rolls up the side of his greenhouse every year, and he's got plants right outside the greenhouse, and he's got flats of cuttings inside the greenhouse, very close to each other, and 2 different years we sampled, and it's not transferring, which is telling us that those macroconidia are not windblown. We also know that it's not seed transmitted. We've done that research. So it's not seed transmitted, and you're likely not moving it around clones if the clones are in the greenhouse, even if the greenhouse is open. All right. So like I said, we have more questions than we do answers. I'm looking at my clock. I have some additional slides here that I can go through if I'm not running out of time. All right. So how do you manage it? We started looking at fungicides. What controls it? What are we going to do in the future? We've got to do something. So there are a lot of ways to test fungicides. Okay, you don't just walk out and do a field trial. It's just too big. So we started with lab analyses, and I'm going to show you some lab analyses. I need you, though, to remember that this is preliminary. This is not the end-all, be-all. These are just some different ways to check some of these. We did what we call poison plate assays, where we put fungicides inside agar, and we put the fungus on, and we saw if it grew or not. We measured the colony size. Another one we did is we co-mixed fungal spores with fungicides. We exposed them for different amounts of time, rinse them off, put them on agar plates. Did they survive or not? That's how we know. That's how we look at like if a fungicide is a cell degrader, if that exposure would degrade. And then the third one we did is some inhibition. We saw if there was any antagonism going on. We have some detached leaf assays pending. I don't think we're going to be doing any whole plant or field studies at this time. Just takes a lot of manpower. All right, so here's the one where we put the fungicide in the media. That would be something like an inorganic type product. And by inorganic, I mean it's something that's not alive. Okay? Now you'll see that in this, so control means there was nothing. I didn't put fungicide in the media. Some of these others, those are synthetic fungicides that are not labeled for hemp. They were just our comparison. So don't think that the best one is one you can go out and use in your fields. And the colony diameter, so on your, do I have a pointer here?
Yes.
I can't see now, the lights are off. Let's see. No? There we go. Colony diameter in millimeters. I just want you to look broadly, okay? We're not looking at nitty-gritty, but I want you to look broadly, is that things like Bravo, which is chlorothalonil, so you got your synthetics through here, Millstop, Trilogy, so as we go down, Sil Matrix. So we had some management with Millstop, for instance, worked pretty good in an agar plate.
Is that organic?
What? Millstop? Millstop is a biological. I'm not sure if it's got an OMRI certification, but Millstop is yes. It's just Cueva is a copper, but from Custodia through Bravo plus Incognito, those are not. All right, then we did this co-mix, as we call it. We mixed up some fungal spores and mycelia with our fungicides. We exposed them. 0 hour is actually about 5 minutes. 2 hours, 4 hours. Then we rinsed them off and we plated them out. We saw how many colonies formed. That tells us, what about these cell-degrading type products? Okay, so that's how we looked at those. We looked at colony forming units. That's just how many grew. Okay, look at it from a broad standpoint. If we didn't use any fungicide at all, we had a lot of CFUs. But cease, double nickel. What are those products? Anybody use those? Those are your bacillus products, right? Those are alive. Sustain here. Sustain is a compost tea. But think about when you've got something that's alive and then you plate it out, it's hard to differentiate between the product and the fungal spores, so those were a little bit harder to count. But we did see that there's some potential with some of these biologicals. And then finally, this is what we call an inhibition study. So if you can bear with me just a little bit, I'll tell you what you're looking at. So we put the 2 fungal plugs right on each end. My time up? 2 fungal plugs on each end. We waited either the same time, we put the fungicide across, or we waited 4 days, let the plugs grow, and then we put the fungicide. When I have a control, that means there's no fungicide at all. This is an example. This is double nickel. So you'll see that that's a bacillus bacterium, so it's a beneficial bacterium, and it is impeding the growth here. So that's how we did that. But basically, we're seeing that double nickel, cease, stargus, those are all bacillus products, were excellent for management of this fungus. So that's good. We have some potential. We have some way to go. Eucladium, which is a competitor, wasn't so good, but Root Shield is a trichoderma. It did pretty good. So these are some potentially that we would take out into the field when and if we do some field trials. All right, so remember, these are lab assays. It's not even detached leaf assays. This is not information you're bringing back to your field yet. We may be doing some field trials, and then recommendations would come after that. All right, so in summary, we still are working on this, and we're going to be working on this for a long time. The more answers we have, the more questions we come up with. It's really complex. So I know I've been promising some good answers for a lot of years, but this bipolaris pathogen, if you see this, by the way, we would be very happy to have samples from your fields. All right, my time up? Time for questions? We have time for questions.
There was 6,000 acres grown last year. Do you know how many acres were actually affected by this?
Okay, his question was, do I know how many acres were affected by this disease? I do not. I do not. A lot of times if I visit a field, I don't even ask how many total acres are there. Other questions? Yes.
One comment and one question someone asked about Bill Scott.
Yes.
Whether under my breath, no, it's not, but it is OMRI listed.
It is OMRI listed?
With restrictions. So if you have practiced all of the management and cultural controls to comply with the organic program, it is useful as a last resort.
Okay, so did you hear that? Millstop is OMRI listed if you meet as a last resort.
Everything else.
Yeah. And this is the organics guy, so this is the guy you would ask for those things.
The question is, having seen this for a few years now, are there any varietal phenotypic characteristics in hemp that may provide some resistance to this pathogen?
Okay, so his question is, do we see any phenotypic resistance? There's so many different varieties that come in and out right now. So we've seen a few, and we've done some screening. I'd rather not show that right now because I don't want anybody to think that I'm, calling out. But there's a little bit of difference. Overall, though, everything we've seen is susceptible, at least to moderate levels. Yeah, so this is a really aggressive fungus. Yes? Yes?
What are some other diseases that you guys have run into that could be like big problems to look out for?
Okay, his question was, what other diseases are we seeing? In 30 minutes, I'm going to tell you exactly that, because my next talk is a diagnostic rundown. Yes?
I was just going to note that we see this on pretty much everything we grow at the end of every growing season.
All plants.
All hemp. We see hemp leaf spot. When that plant reaches maturity, we see it on pretty much every fiber, grain, whatever. We see it.
And two, there's septoria leaf spot, cercospora leaf spot, alternaria leaf spot that look almost identical. So we need to confirm. So just because it's a leaf spot doesn't mean it, but we do see a lot of this bipolaris.
One more question.
Yes.
What about dipel? Is that considered a bacillus organic or not?
Dipel? I don't know. I don't know dipel. Sean, dipel?
I'd have to look it up on Omni's.
Okay. Okay. I can look it up during break. How about that? I'll let you know. Okay.
Join me in thanking Dr. Gauthier.